All right, so we're going to kind of review cellular respiration here by looking at this case study. I'm going to pass around these little slips that have a bunch of questions. We're going to go part by part. So this is kind of the intro. I'll let you read it while I pass this around. I'll move into part one. You'll answer some questions. We'll come back together, so on and so forth. So while I'm passing this around, go ahead and read that.
I didn't do that exactly. I just did sprints on my own. So I just did 5 killers and then the 6. No, I never will. I never will. No, I never will.
Oh, thank goodness, there's no room to write.
It's all discussion. Yes.
Yeah. Okay. So, yeah, I agree. I'm not ready for the 400s on Friday. Oh,
Whoa.
All right, did everybody get a question sheet? All right, I think so. All right, so I'm going to bring your attention back here once more. So we're looking at a case study. We are imagining that we work as a medical examiner and that there have been a variety of deaths in the past few days, and we want to know what's going on. All right, so we're going to examine the data and determine the deaths of these victims. So, part one, there is a lot of text. I apologize. I didn't want to print you out a whole 5-page packet. So I broke it down to part one. Go ahead and read through this and then answer the questions pertaining to part one on your slip, and then we'll come back together. Feel free to work in groups. I'm going to pass around your in-class assignment from Friday.
Bro, people drop some.
Oh my gosh, I'm also telling you. Why are we taking Tylenol? Your boss calls me
I think it's better. I didn't want to go No, I'm definitely still recovering. I definitely am not fast. But I will run for you. Yeah. It's so cute.
All right, does anybody need more time on these questions? All right, let's come together and discuss these two questions. So the first question, are there any similarities or connections? What do we think? Yes, all right. And what are those similarities? Tylenol, yes. Same neighborhood. Similar symptoms. Collapsing, yes, part of those similar symptoms. Anything else? Dying very quickly. Yeah, so there's a lot of similarities. So what questions would you ask their families to try to figure out what happened?
Let me see that bottle.
How much they take. Let me look at the Tylenol.
Do they normally take Tylenol?
If they normally take Tylenol.
I've never had that before. Like,
Yeah.
had like a bad reaction.
So like their medical history would be a good thing to ask.
Maybe if they like normally complain of sicknesses, or if they just kind of suck it up and go about their day.
Yeah, do they normally say something about their illnesses? Any other questions we might want to ask?
Could there be something totally unrelated to that, like the water or something?
It could be. We could ask them other questions about their daily routine and what they did. Anything else? Yeah, history of their health conditions. Right. All right. So we have some good questions to ask the families. So in your opinion, are these deaths connected? All right, from what we're given, yeah, we could say that they might be connected, totally. Or it could be something totally random, like we said before. It could be something in the water. All right, but it looks like these are probably connected. All right, and I'd ask you the why or why not, but we just kind of mentioned that. They all took Tylenol. They all had the same symptoms, all in the same area. All right, so let's move on to part 2 and look at the autopsy report. So again, I'm going to give you some time to read this, time to discuss. I hear a question?
Why is there a high oxygen in blood?
All right, it's getting a little quieter in here. I'm assuming everyone's had a chance to look at the questions. So let's move on to the question about part 2. Let's see here. So, what function of the cells was interrupted in these patients? Thinking about the organelles involved. ATP production, because we know there is a lot of mitochondrial damage. Yeah, totally. That's kind of an easy question there. Could this loss of function lead to the death of these individuals?
Yeah.
How so? Can't get energy, obviously the lack of oxygen. Right, and you need energy to power all these different cellular processes in your body. Without the energy, you literally can't function. Yes, cell death leads to organ death leads to death. Very nice. So given this data, what's a major inconsistency here?
The oxygen levels in the patient's blood were higher than normal, but hypoxia means that they died from lack of oxygen.
Yeah, so there's definitely some inconsistency here. They died from lack of oxygen, but they had plenty of oxygen in their blood. All right, so what's going on? Let's look at the detailed analysis. All right, again, there's some questions involving this part.
Sorry, I switched. Yes. I think that was really good. So we're in Oh, I sit next to someone left-handed. I swear.
You're so persecuted. I know.
Actually, I'm kind of left-handed.
You know what sucks writing in a binder left-handed? Terrible.
Then why don't you just take the paper out?
Because
It's
that's
binder.
inconvenient.
I'm kind of left-handed. I hit the volleyball left-handed.
Does that matter what side? Like, you like stick your outside of it? Like outside
You're
or
not
right side?
right-handed. You're outside hitter.
Like your right side of yours doesn't matter?
No, if you're a good hitter, you're somebody.
Okay, I was just curious.
Like normally a lefty person. Does that make sense?
Because you want the inside hand?
Yes.
So you can, okay, that's fair. Yeah.
Sometimes I write people hitters right side.
You do your. Okay, that's enough.
That's not bad. Could be better. You should right-handed?
Random tweak your shower a little bit?
It is not on. It was like, I'm done. It is never
All right, how are we doing on the questions? Do you need more time? Because I know these are more complex. All right, seems like we're still discussing a little bit. I'll give you a few more minutes.
Oh, this is your guys' stuff.
Coming up with that hypothesis over here. All right. That's a good one. Huh? So, yeah, I think there's something going on there. Oh, you got to operate in the cast. How long do you have to have the cast on? Your right hand, right? Well, I know. My sister, when she was younger, broke her left arm. Her right with her right hand. Over 6 weeks. Yeah. Relative to hypothesis. This is just you can cut up and put it when you're done. You bring up the height and come up.
Where is your
If it isn't able to do that.
Yes, it is. Do you know where the biology office is?
I can find it.
Okay. So it's on this floor, down older part of the building. I guess it's not this floor. It's on the second floor.
Okay.
You'll see it right in the main hallway there, and then there's like this little alcove where there's like this hallway.
Okay.
It's kind of behind some seats.
Okay.
That's where my office is.
Okay. Can I come tomorrow?
Totally. Yeah. If my door's like prop closed, just not.
Okay.
Yeah. I normally have it prop closed. Over there.
Yeah.
Take my mask off.
Okay, awesome. Thank you.
All right. Let's come back together. So we're looking at our detailed analysis here. Let's see, what are the roles of these metabolites in metabolism? All right, let's look at glucose. What's the role there? It makes pyruvate. Yeah, it's the first step here. So what's the role of pyruvate? Acetyl-CoA, yeah. And in the process of making pyruvate, we're making a little bit of energy. What about NAD plus and NADH? What do they serve as? Yeah, so they're kind of like the electron carrier molecule. Yep, they provide those electrons in the electron transport chain. All right, so is glucose a substrate or a product? Substrate, yeah, exactly. It's a substrate. We only see it once. It's being broken down, and then it's no more. All right, what about pyruvate? Product and a substrate, yes. So it's a product of glycolysis, but it's being used as a substrate to make acetyl-CoA. What about NAD plus and NADH? Substrate and product, yes. They're going to be oxidized and reduced throughout this whole series here. So they can be used as both a substrate and a product. All right, let's see. So are there any abnormalities that we see here? Yes. Yes, all right, what are they? Yeah, too much NADH, not enough NAD+. All right, that kind of really jumps out there. It's way different. So what might be a hypothesis about what pathway in cellular metabolism is affected? All right, and what's your reasoning? Yeah, great hypothesis. So we have a ton of NADH. We know that's produced in all the previous steps, and then in the electron transport chain, that will get oxidized into NAD+, which we don't have a lot of. That's a great hypothesis. Anybody else have one? All right, sounds good. So let's take it a step forward here and look even further into our analysis. And I included a little picture here because it's going to talk about cytochrome C. That is very specifically this protein embedded in the membrane. All right, let's come back together here. So, what effect would cyanide have on the electron transport chain and the production of ATP?
It prevents the electrons to transfer to oxygen, which renders the body unable to derive energy from the oxygen.
Yeah, so it prevents the transfer of electrons to oxygen, right? So that's not happening anymore. And indirectly, that affects the proton pump, so we're not pumping out those protons, setting up that gradient, and that's preventing ATP synthase from being able to work. Any questions on that? Because I think that was a good answer. All right. So if we had given them... Oh, question 2, I'm jumping ahead. So given what you know about the action of cyanide on cellular respiration, why would these patients have died from lack of oxygen, even though their oxygen levels were normal, if not a little elevated?
Kind of like backing up the oxygen, so the oxygen can't really flow through to do what it needs to do, and it just gets all backed up.
Yeah, we might have plenty of oxygen, all right, but because cyanide's preventing those electrons being transferred to the oxygen is not being utilized, all right, so it's backing everything up. All right, so if we had given them artificial respiration, done CPR, given them extra oxygen, would this have made a difference? No. All right, they already had oxygen. They don't need more oxygen. So what might be the source of this poisoning? Tylenol. Yeah, so everybody had taken Tylenol. It's kind of the common denominator there. And so how should public health officials and police respond? What should we do? Take it off the shelves. Recall the product. Yes. So this is actually what happened. So this is based off a true story, the cyanide murders in Chicago. Somebody tampered with Tylenol, put cyanide in the tablets instead, and a bunch of people died, and they recalled the product and figured out what was going on. This actually happened.
That's so sad.
So this led to this whole effort in preventing tampering with medicines. So you have, it's kind of weird to say, but you have this to thank for this not happening in the future. But let's go back here, because I actually have a video that's going to show exactly how cyanide plays a role in cellular respiration. And I think I figured out the audio issue, so we are going to find out.
So by now you've seen this graphic quite a few times.
Maybe I did not.
His name is Andy. Look at his blue light glasses.
What's his name is
No.
Tim. Yes.
Gray.
All right, let's try that.
To one of the
Yeah.
proteins here, giving up protons and electrons, protons being pumped across the membrane as the electrons are passed down the chain, eventually to their final electron acceptor, which is oxygen. Pumping of those protons creates a gradient so that they can flow back down through ATP synthase and be used to synthesize ATP. Here we're being told that cyanide irreversibly inhibits cytochrome C oxidase, which is this protein right here. So this diagram actually has a little thing where we can see directly the result of cyanide. So we're going to add cyanide to the system. Here comes cyanide. It's binding to the cytochrome C oxidase. Electrons cannot be transferred now from this protein to the oxygen. That oxygen is blocked. That also means that electrons cannot be transferred from this protein to the cytochrome C oxidase. They can't be transferred from this protein to this protein, from this protein to this protein, from this protein to this protein. That also means that this protein at the beginning of the chain can no longer remove electrons from NADH. And this is why the NADH amounts have risen dramatically, and also why the NAD plus amounts are reduced. Because that reaction by which NADH is oxidized to NAD plus can no longer occur. It's sort of like a traffic jam. You've blocked the off-ramp, and the whole road has backed up. And if you think of this as sort of the on-ramp for electrons, no new electrons can make it into the system. No new NADH can be oxidized. Of course, the oxidation of NADH is just a means to an end, which is the generation of ATP. The real problem here is that by shutting down the electron transport chain, we are no longer pumping any protons across this inner mitochondrial membrane. That means we're not setting up a gradient across that membrane. That means there is no process that's going to drive protons down through the ATP synthase to generate ATP. This means a failure of ATP production and death. This is, at a molecular level, what is happening when a person suffers from cyanide poisoning. Depending on the dosage, this could be fatal within minutes, or it could be something that a person rushed to a hospital could recover from with some treatment. We'll talk about that treatment in a moment.
All right. So that's exactly how cyanide is inhibiting cellular respiration. I hope that makes it a little bit more clear. All right. And like he was talking about, it depends on dosage, but these individuals, they had a relatively high dosage of cyanide, which is why it occurred very suddenly. All right. So we're going to switch gears now, and I'm going to do a little bit of lecturing on fermentation, but I don't have a ton of slides. Then we're actually going to go back and do another quick case study. All right, so fermentation, anaerobic respiration. All right, now we're talking about energy being produced without oxygen being present. All right, so this can occur in two main pathways, anaerobic respiration and fermentation. All right, so there's plenty of examples in biology of organisms that do this. Bacteria, archaea, some fungi, even our muscle cells will sometimes perform either anaerobic respiration or fermentation. Our muscle cells specifically, fermentation. Especially when we're exercising intensely, we don't have that oxygen to provide as a fuel source to these muscle cells. They will switch to fermentation. All right, so what's the difference between the two? So anaerobic respiration is we're going to use a molecule other than oxygen as our final electron acceptor. All right, so we're still going through cellular metabolism. We're going through glycolysis, breakdown of pyruvate, into the Krebs cycle, electron transport chain, all that's still occurring. We're using a different electron acceptor though. All right, so we're no longer using oxygen. Versus fermentation. So this is where we're producing ATP via substrate level phosphorylation only. All right, fancy way of saying we're going to go through glycolysis and glycolysis only. All right, and with fermentation, there's going to be no net oxidation of any organic molecules. And we'll go over that a little bit more in depth here. So first, let's talk about aerobic versus anaerobic respiration. Right? So far we've been talking about aerobic respiration. We've been talking about the presence of oxygen. That oxygen is that final electron acceptor because it's highly electronegative. Now bacteria and archaea specifically can use other electron acceptors, such as nitrate or sulfate. All right? The example is given up here. So they don't need oxygen. They can use these other electron acceptors instead. Now just kind of a quick side note. We can use other electron donors besides glucose. So some of these organisms, instead of utilizing glucose, they can utilize methane or hydrogen sulfide or even hydrogen gas to get this process going. Right? A little less efficient, slightly different pathways involved, but kind of the same general concept. And then again, all this is happening in the electron transport chain and ATP synthesis. We're still going through that process. Right? We're still having that buildup of the electrochemical gradient across the mitochondrial membrane. So let's look at an example. And this example uses nitrate as an example here as the final electron acceptor. So E. coli, all right, so a bacteria we typically find in our intestinal tract, they utilize nitrate as their final electron acceptor. And they're going to produce a protein instead of the cytochrome C that we just saw in our example with cyanide. They're producing nitrate reductase as their protein. All right, this is going to be the protein that allows the electrons to be accepted by nitrate instead of oxygen. So they're producing a whole other protein, right? And the end result is that nitrate is converted to nitrite. All right, very similar, but different molecules. And this is going to act the same way. We're generating that electrochemical gradient. Right? Now, one quick side note, though. These are not as effective. All right? So they have a lower potential energy than oxygen. So iron can also be used as an electron acceptor, nitrate that we just talked about, and oxygen. Oxygen's really electronegative. All right? So when we go from glucose to oxygen, it's much more efficient than if we go from glucose to nitrate. Right? So this is kind of what this image is telling you very briefly. I'm not going to go too in depth on the whys of that, but just know that oxygen, highly electronegative, works really well as an electron acceptor, which is why we don't utilize nitrate, all right, because we have oxygen. Right? Oxygen's just much more efficient. All right. Switching gears to fermentation here. So fermentation is where some organisms can only use oxygen as their final electron acceptor. They don't have the ability to use nitrate. They can't use sulfate. They can only use oxygen. So when oxygen is limiting, right, we have to do something else. And in fermentation, this something else involves glycolysis and a second step of regenerating that NAD+. All right. So, and I just realized I'm not presenting this, am I? Let's make it a little bigger for you. All right. So one of the problems with fermentation, and I'm just going to go back to this diagram here, is that we have glucose being broken down to pyruvate. We're generating 2 ATPs. All right. We're also oxidizing NAD+, all right, to form this reduced version of NADH. All right. So we're building up a lot of NADH. And that's one of the problems of fermentation, all right, is that we're building up NADH, and this can create some problems. So high concentrations of NAD+ or NADH will haphazardly donate its electrons to other molecules. All right, so this is an electron carrier. It's easily oxidized. It's easily reduced. So it's going to easily be able to donate its electrons to other molecules. But this promotes free radicals, the formation of free radicals. And free radicals are these molecules. They're highly reactive. they can damage DNA, and they can damage cellular proteins. All right, so we don't want free radicals produced. And finally, if we have a buildup of NADH, all right, that means we're decreasing NAD+, which is what we need for glycolysis in order to go through glycolysis. All right, we got to keep glycolysis running. So, a couple of ways that we deal with this issue. The first is lactic acid formation. And this is what our muscle cells will go through. All right, they'll build up this lactic acid. So pyruvate is going to be reduced to make lactate. All right, so we're still going through glycolysis. That's step 1. Step 2, this pyruvate is reduced to make lactate. So if we're reducing lactate, we're using that NADH that we built up in glycolysis. All right, we are oxidizing that back to NAD+. All right, now we have NAD+. Now we can go back and go through glycolysis again. All right, so that's one way that we can kind of get rid of that problem of too much NADH. Now with lactate, once there is sufficient oxygen, that lactate can be used to build pyruvate molecules and go back through this step or go back through cellular respiration once there is oxygen. Sometimes lactate will be used to build up other glucose molecules within the body. So this lactate is used in other areas. All right, and we utilize this by making cheese. All right, this is a common way to make cheese, is to utilize these bacteria that make lactate. Now the other method is alcohol fermentation. All right, so this is very common in yeast. So yeast can use oxygen when it's present, and when it's not present, it goes through this alcohol fermentation. Now because it can use both pathways, it's a facilitative anaerobe. All right, and in lab this week, we're going to be looking at yeast cells specifically, so we'll be looking at this process. But essentially, we're going through glycolysis, all right, creating that 2 ATP, creating these pyruvate molecules that will be broken down into ethanol. In the process of doing that, we're getting CO2 as a byproduct, all right, and regenerating NAD+. So we can go back through and do glycolysis all over again. All right. Now this does have a middle step in it that is not shown in this picture. Pyruvate's going to be broken down to acetaldehyde before it reaches alcohol. All right, so it does have an intermediate step there. Now we use this in making wine and in making beer. All right, we utilize this process. This process can be reversed essentially. All right, so if we have the presence of oxygen suddenly, I put this little diagram at the bottom that ethanol can turn back into acetaldehyde, can turn into acetate, can turn into acetyl-CoA, and go through the rest of cellular respiration. All right, so just as a review of the fermentation, all right, we're breaking down organic molecules to harness energy without any net oxidation. All right. So what that means is this NADH is constantly being regenerated. All right, we're not building up NADH at any point. All right, electrons are removed from glucose, and then the electrons are going to be donated back via lactate or ethanol production. So that NADH made during glycolysis cannot be used to make more ATP. All right, this is just kind of where it stops. We go through glycolysis, we stop. All right, so the result is we can only make 2 ATP in this pathway. That's not a lot of ATP. All right, now because these organisms that typically rely on fermentation, such as yeast, they're typically single-celled. All right, they're pretty small. They don't have huge energy demands like us. All right, so they don't need a ton of ATP, so fermentation will do okay for them. All right, it's not as efficient, but it will work. All right, so I also have this case study. We only have 3 minutes left. I'm going to leave this up here. Read this before Wednesday, and we will pick up on that. Bring this sheet back because it has the questions for this case study on the back. It's going to be a relatively short case study. I do have some additional resources here. So this is a BuzzFeed video on the cyanide murders, in case you're interested in learning more. And this case study we're going to do next is also a true story. If you want to look ahead, go ahead and read this NPR article. There is an assignment 2 due on October 18th. All right, that assignment is up on Moodle. We have not gone through everything on that assignment yet. So you can start working on it, but you won't be able to answer any questions about photosynthesis yet. So it's up there if you want to work ahead. And then again, quiz 3 on Friday.